The overall goal of the following experiment is to illustrate the use of glycosidases for the enzymatic deglycosylation and analysis of N and O-linked glycosylation on a model glycoprotein. This is achieved by treating a glycoprotein with PNGase F or with the protein deglycosylation mix. Also, the protein deglycosylation mix was supplemented with a mixture of additional exoglycosidases, which sometimes help remove otherwise resistant sugars. We illustrate this method using a model glycoprotein, recombinant human chorionic gonadotropin beta, or HCG beta. Next, SDS-PAGE, followed by Kumasi blue staining and a sugar-specific staining method, ProQ Emerald 300, are used in order to analyze the deglycosylated HCG beta sample. Results are obtained which show that HCG beta is heterogeneously glycosylated and contains multiple glycoforms based on the differences in protein migration with and without glycosidase treatment and the diminished signal on ProQ staining as the glycans are successively removed.
The main advantage of this technique over existing methods such as mass spectrometry analysis is that it's simple, uses common laboratory equipment, and is good for the novice glycobiologist. This method can answer key questions in the glycobiology field, such as, is my protein glycosylated?
To begin enzymatic deglycosylation, thaw the supplied buffers from the protein deglycosylation kit. Mix the tubes thoroughly and keep at room temperature.